Good morning, my dear student. Uh, Here I'm going to discuss bilirubin metabolism and jaundice. Before going to start this topic, I would like to discuss some basic things that would be the pathophysiological importance of bilirubin metabolism. The bilirubin is the end product of heme degradation. Serum bilirubin level is an important clinical marker of hepatobiliary excretory functions. Hepatic, uptake, storage, conjugation, and excretion of bilirubin are finely balanced. Therefore, enhancement of bilirubin throughout require coordinated induction of multiple genes which may be mediated by nuclear receptor. Then we come to the source of bilirubin. There are two sources of bilirubin. One is erythroid source and other one is non-erythroid source. In erythroid sources, the normal means in physiological condition there are the senescent erythrocytes means the old RBC and the free heme. The second one is abnormal like hemolysis. In hemolysis, there may be extravascular and intravascular. Then the ineffective erythropoiesis. Non-erythroids, the cytochromes, catalase, peroxidase, tryptophan paralase, and myoglobin. Now you see the old dying RBC, that is the mononuclear phagocytic systems, where the degradation of RBC takes place. The one is the liver, spleen, and bone marrow, and the most of this destruction of RBC takes place in the spleen. Then from the RBC, heme comes out, and this heme converted into biliverdin by the action of microsomal heme oxygenase. During this process, the carbon monoxide is released, which is excreted via the lung, and the iron which are released, which reutilized for the production of RBC. Then this biliverdin is converted into biliverdin bilirubin with the help of biliverdin reductase. Then this bilirubin goes to the blood circulation. This bilirubin is bound to the albumin, and albumin bound bilirubin goes to the liver where the conjugation takes place, and then conjugated bilirubin is excreted, which is stored in the gallbladder. Similar slides here. Here the RBC is broken down in the heme and globin. You see this biliverdin is by the action of biliverdin reductase. You see this bilirubin and this bilirubin. But here in the liver, you see this ligandine attached to the bilirubin, and this bilirubin, the UDPGA, uridine diphosphate glucurylin transferase enzyme, is where the glucuronidation, which is responsible for the glucuronidation, or you can say it's a conjugation. So this glucuronyl transferase enzyme is basically a smooth endoplasmic reticulum enzyme, which is present in the smooth endoplasmic reticulum. Now the bilirubin monoglucuronide, then the bilirubin diglucuronides are also for most of this is a bilirubin diglucuronide. Most of the time there is a bilirubin diglucuronides are formed, and the conjugated bilirubin, which is yellow to green in color, these conjugated bilirubin is basically the conjugations takes place. This goes to the bile, and when conjugated bilirubin goes into the intestine, where the beta glucuronidase enzyme acts on the bilirubin, which converts it to the urobilinogen. This urobilinogen, the 90% recycled by the liver intrahepatic circulation, some are reabsorbed, 10% goes in urine, and some are goes to the feces. Now, here this is nothing but the structural, how the structures of heme and how this, what is the structure of bilirubin. Here the same thing. Here heme oxygenation acts, then biliverdin reductase acts. So it is nothing but the similar, the repetition of the same. The linear structure of bilirubin, where you see the two dipyrrole joined by the central methane bridge. Again, the bilirubin contains several polar groups. It is it. It is insoluble in water. So conjugation with glucuronic acids makes bilirubin water soluble. The internal hydrogen bonds of bilirubins are disrupted by the conjugation of propionic acids, carboxyl group, with glucuronic acids. Internal hydrogen bonds of bilirubins are disrupted by the conjugation of propionic acid, carboxylate with glucuronic acids. Now the phototherapy changes the configuration of bilirubin, making it transiently water soluble. This the phototherapy. Then the question comes to what is the phototherapy? The phototherapy is nothing but it is a treatment of choice of neonatal jaundice. And this phototherapy, we use a particular wavelength of light, and that change there is a photoisomerization of bilirubin, which changes the bilirubin into lamirubin, which becomes transiently water soluble, and it is excreted out from the body. Then there are the 3 or 4 slides which depicts the how this bilirubin, albumin bound bilirubin comes in the liver and what happens. So the first is you see the sinusoidal surface of the liver, you see the canicular surface of the liver. Here you see the tight junction. Here you see the liver sinusoids, and here you see the fenestrated endothelium. Then you see the bilirubin circulate bound to serum albumin, which previously we discussed. Then what are the significance of albumin binding to the bilirubin? The significance are it keeps bilirubin soluble, it prevents tissue deposition of bilirubin, it prevents renal excretion of bilirubin, and the other significance of that, there are certain drugs that displace bilirubin from albumin may precipitate kernicterus. And these drugs are the 2 common drugs, but which is not common. Sulfonamide is a common antibiotic which we used previously, but still we are using these drugs. Then coumadins are another group of drug which which displace the bilirubin from the albumin. So if we use this sulfonamide and coumadin groups of drug, it may precipitate kernicterus, particularly in the children or neonates. Then the bilirubin circulate bound to albumin. Yes, here you see at the sinusoidal surface of hepatocyte, it dissociates from the albumin. Next, the albumin goes. Next, you see the bilirubin enters into the hepatic sinusoids, probably by facilitated diffusion. Next, you see the bilirubin enters to the sinusoidal surface probably by the facilitated uptake is energy independent and bidirectional. This is very, very important. Now, it is bidirectional, means bilirubin enters into the hepatocytes and hepatic sinusoids and can exit from the hepatic sinusoids. So what does it mean? It means there are certain things which bilirubin so what are the this is one step. This is the important step where if bilirubin has to be conjugated, this bilirubin uptake process is very, very important. And what are the factors which reduce this uptake of bilirubin in the hepatic sinusoids. The one in neonates, this uptake is in problem, which is a not a pathological, but it's a physiological condition. In cirrhosis of liver, where there is a certain the fibrosis of liver and the inflammations of liver takes place, in that condition, bilirubin uptake is reduced. The third thing, there are certain drugs which can reduce the bilirubin uptake, that is novobiocin. In some cases of Gilbert syndrome, which I will discuss later on, that what is the Gilbert syndrome. Now, here you see inside the hepatocyte, bilirubin bind to the cytosolic protein termed ligandine. Understand? So when the bilirubin enters into the hepatocytes, there is a ligandine, bilirubin attached to the ligandine, which are the same as glutathione S transferase, Okay, so GST binding inhibits the efflux of bilirubin, thereby increasing its net uptake. So GST binding inhibits. So this binding of bilirubin to this ligandine is responsible for the net efflux of bilirubin, inhibits the efflux of bilirubin, thereby increasing the net uptake. Understand? If there is no ligandine-like structure which is present in the hepatic sinusoids, then because the bilirubin uptake is a bidirectional, so there would be no net influx. So for that purpose, here ligandine, which are the same as glutathione S transferase, is very, very important. Now you see the conjugation of bilirubin with glucuronic acid is catalyzed by the uridine glucuronyl transferase 1A1, which transfer glucuronic acid from the UDP glucuronic acid to bilirubin. Understand? So this process, this is very, very important. And here you see the reactions, how the reaction takes place. Okay, you see the conjugation with the glucuronic acid makes bilirubin water soluble and non-toxic. Glucuronidation is essential for biliary excretion of bilirubin. It is very, very essential step in bilirubin metabolism. Now you see through this chart the same thing. RBC after 120 days in lifespan, breakdown by the reticuloendothelial system of the liver and spleen, particularly the spleen. This hemoglobin, the protein part is recycled, iron part is recycled. It converted into protoporphyrin. This bilirubin is conjugated with is bound with the albumin. This bilirubin comes in the liver, conjugated bilirubin goes into the intestine. This urobilinogen is basically converted into the other types of that, urobilin, stercobilin, and mesobilin. There are other types. And here the small amount passes through the kidney in urine, urobilinogen, which is finally converted into urobilin, and the urobilin in the stool, which is basically a stercobilin. Okay, the same thing is in this slide. Nothing new. Almost the same thing here. Stressing on that. You see the same thing, but here the one thing which is highlighted, the urobilinogen is converted into the stercobilinogen, mesobilinogen, and urobilinogen. And in large intestine, this stercobilinogen converts in stercobilin, and this urobilinogen is converted in urobilin. So urobilin is responsible for the yellowish coloration of urine, and this stercobilin is responsible for the yellowish coloration of the stool. Now this UDP glucuronyl transferase, there is a substrate, and this UDP glucuronyl transferase where the glucuronides, and this is very, very important. The UGTR ER proteins that convert many internal and exogenous toxin to non-toxic metabolites. So this UGT enzyme, uridine glucuronyl transferase, is not only responsible for the glucuronidation of this bilirubin, but is also responsible for the glucuronidations of different exogenous toxin to non-toxic metabolite. And you must know that the liver is a primary site for detoxification process goes on in our body. So liver is mostly responsible for the detoxification of all the exogenous products. What most of the exogenous products are detoxified and converted into more harmful to the less harmful substance by using the different kinds of enzymes. Okay. Now, the UGT family of enzyme concentrated in the liver. Here, the one UGT isoform UGT1A1 conjugates bilirubin as an essential for its secretion. So, it may comes as a multiple choice question that which form of UGT, which isoform of UGT is responsible for the conjugation of bilirubin. So, this is the UGT1A1. Okay, so understand. Then inherited UGT-A1 deficiency, one called jaundice, because if there is a no conjugation or the less conjugation, then the unconjugated the result is unconjugated hyperbilirubinemia. Understand? Now the second part of this topic is jaundice. So the yellow color of skin, nail beds, sclera, caused by deposition of bilirubin secondary to increased bilirubin level in blood is known as jaundice, or which is also known as hyperbilirubinemia. So jaundice is not a disease, however, it is a sign of an underlying disease. Yes, understand there. You should know. You must know this fact that the jaundice is a manifestation of the various disease, various liver disease in which the jaundice appears. So jaundice is not a disease; it is a sign which the doctor elicits when he or she observe the patient. Okay. Now the classification of jaundice. This is the most accepted classification of jaundice. There are other way to classify the jaundice, but the most accepted or the common the most accepted way to classify the jaundice is this. The one is prehepatic, hepatic, and the post-hepatic. So then prehepatic, what is this? The overproduction of impaired over. There is two possibility of prehepatic jaundice. The cause of the prehepatic jaundice is two. One is overproduction of bilirubin, and other one is impaired uptake by the liver. The second is hepatic. In hepatic, whatever the cause, that cause is the decreased conjugation. In post-hepatic jaundice, whatever the cause is decreased excretion, that is intra or extra hepatic obstructions. The obstructions may be intrahepatic or the obstruction may be extrahepatic. The same thing, the type of jaundice, prehepatic, the commonest cause of prehepatic is hemolytic anemia. Hemolytic anemia may be of different types. Yes, in hepatic, the first is hepatitis. The hepatitis may be viral hepatitis, and there are other kind of hepatitis, and the other kind of hepatitis is autoimmune hepatitis. Then cirrhosis. The cirrhosis is a consequences of this hepatitis, and which is actually the next level of hepatitis. When there is infections by the hepatitis B, hepatitis C, or the autoimmune hepatitis, all the alcoholic hepatitis, this may lead to the first is a fatty liver, then the cirrhosis, and finally there is the chronic liver disease, and finally there is a end-stage liver disease. So that is the second one. In hepatic jaundice, the Crigler-Najjar syndrome and Dubin-Johnson syndrome, which is an inherited form of jaundice, which I will discuss later on. Then the Rotor syndrome, the three inherited form of jaundice, that is Crigler-Najjar, Dubin-Johnson, and Rotor. I will discuss later on. The next one is a post-hepatic. In the post-hepatic, the gallstone is the commonest. Then the malignancy of the gallbladder, malignancy of the pancreas are there, and there may be inflammations around this, around the gallbladder, around the pancreas, like acute pancreatitis, the chronic pancreatitis, that may be a cause of jaundice. Here you see the prehepatic jaundice. What the cause? Due to increased RBC breakdown. The same thing I'm again discussing. The rate of RBC lysis and bilirubin production more than the ability of liver to convert to the conjugated form. Understand? Means the production of RBC, the rate of production of destruction of RBC is much higher than the ability to uptake and ability to conjugate. And in that way, the prehepatic jaundice appears. So what are the causes of prehepatic? That one is erythroblastosis fetalis. You know, the mismatch RH transfusions. Then hemolytic anemia, the types of hemolytic anemia. There are several types of hemolytic anemia. And the third one is transfusion reaction, which is a mismatched transfusion reaction. So in the prehepatic jaundice, the types of bilirubin is what? You understand? You know that the indirect bilirubin is more than direct bilirubin. Indirect bilirubin means unconjugated bilirubin is more than the conjugated bilirubin. Why? Because the production of bilirubin are there, but the conjugations are not there. Why? Because the uptake is in problem, or the excess amount of RBC destruction which overloads, or which is where the conjugation capacity is not matching the production of or the destruction of RBC. So the conformational test is what are the conformational tests? That one is potassium is high, serum potassium is high, because if the RBC lysis takes place, the serum potassium level goes up. Then the CBC, where the incomplete blood count, you will see the low hemoglobin is another test which gives you the idea that it may be a hemolytic jaundice or prehepatic jaundice. There are certain other tests also which can help to determine the nature of jaundice, which is whether it is a prehepatic, hepatic, or posthepatic. Then the hepatic jaundice, which is also known as hepatocellular jaundice, due to liver cell damage. Now, most of whatever the cause is the damage of liver cell. That's why it is known as hepatocellular jaundice. Then the conjugation of bilirubin decreases. Here the indirect bilirubin level is high because the production is production is normal, uptake is normal. The problem in the conjugation. So in the conjugation is not taking place, so the unconjugated bilirubin level is high, and that is known as unconjugated hyperbilirubinemia. So the bilirubin that is conjugated not efficiently secreted into bile but leaks to the blood. Maybe there another cause, that is the direct bilirubin also increases, but most of the time there is both the two process, whether one is conjugation is in problem and other one the conjugated bilirubin is not efficiently secreted into bile. Then the causes are, one is cirrhosis, which is very very common. The cause of cirrhosis may be different. The cause of cirrhosis may be the viral hepatitis, maybe alcoholic hepatitis, maybe autoimmune hepatitis. Okay, then the hepatitis itself, then the Gilbert disease, which is a type of inherited hyperbilirubinemia. Here the type of bilirubin, direct bilirubin, indirect bilirubin, total bilirubin, all are high. Okay, so in this condition, hepatocellular condition, direct bilirubin means conjugated bilirubin level is high, in unconjugated bilirubin level is high, total bilirubin, all are high. So what are the other tests which you can give you the certain clue regarding this hepatic jaundice, that is ALT and ST, the SGPT and SGOT are also high in this. Then what are in the obstructive jaundice or the post-hepatic jaundice, due to obstruction of bile duct which prevent passage of bilirubin into the intestine. Here the direct bilirubin will back to liver and then to circulation, elevating its level in blood and urine. Here the conjugation, the production, the destruction of RBC, rate of destruction of RBC is normal, uptake is normal, the conjugation is normal, but the problem in the excretion of the conjugated bilirubin, and that's why the direct bilirubin level goes up and the conjugated bilirubin level go or the conjugated bilirubin level goes up. And as the level of conjugated bilirubin goes up, it goes back to the liver, will back to the liver and then to circulation, and it elevates the level. The cause is one is biliary stricture, the cancer of pancreas or gallbladder, and the gallstone. Here the bilirubin is only the high is direct bilirubin. So this is a conjugated hyperbilirubinemia. And what are the one important test which gives you the idea whether it is the hemolytic, pre-hepatic, hepatic, and post-hepatic. The one is alkaline phosphatase that is very high in this post-hepatic jaundice. Something like that, similar thing you can see here. The only thing which I would like to highlight, that is the Vandenberg test. The Vandenberg test is based on the dye where the indirect positive means the unconjugated bilirubin is present. Direct is positive means the conjugated bilirubin is positive. Biphasic means the direct and indirect both positive. Okay. And the second thing which I would like to highlight, the urine color. Urine color is acoluric, dark color and coluric. This normal or acoluric. This is not very important, but the important is the stool color in the post hepatic is clay colored. This is very significant finding, which the patient tells sometimes that I have the clay colored stool or almost the light colored stool. That gives you the certain idea about the type of jaundice. Then the alkaline phosphatase level in the post hepatic. See, the alkaline phosphatase level in the post hepatic. Alkaline level in post hepatic. You see the post hepatic that which is a 10 to 12 times than the normal. So it is very important. And in urine, the pre-hepatic, the conjugated bilirubin is absent. Here, the conjugated bilirubin in hepatic, it is present. In the post-hepatic, the conjugated bilirubin is present and high. In urobilinogen, the pre-hepatic, this is the present urine urobilinogen is present. It in hepatic present, may be present or may be absent. In the post-hepatic, the urine urobilinogen is absent, which is very important. And the bile salt in pre-hepatic, urine bile salt absent and urine bile salt in hepatic present, but in post-hepatic it is markedly increased. Level of the urine bile salt is markedly increased in post-hepatic. Now here I would like to discuss the patient appears jaundice. First, it's a very clinical thing which you should at least you go through this very 2 minutes or 3 minutes slide that will help you in the later part of your life or later part of your clinical life. Here, the first is perform history and physical examination. Any patient comes in your clinic or in your outdoor. First, you take history, then the physical examination. And in physical examination, what is important, which I discussed in the practical class, that the icterus comes in the general examination, in the physical examination, when where you see the what the upper bulbar conjunctiva. What you look for the upper bulbar conjunctiva. If the upper bulbar conjunctiva is yellowish in color, that is the sign of the jaundice. Then you draw some screening lab test like the direct bilirubin, indirect bilirubin, the complete blood count, then the liver function test. In that liver function test, here it is the alanine transaminase, aspartate transaminase like SGPT and SGOT, alkaline phosphatase, gamma glutamyl transferase GGT, prothrombin time, and international normalized ratio, that is albumin and proteins. Then the lab studies. If the lab studies are normal, Consider other causes, and in other causes, the skin colorations, what the was spray tanning, which may the carotenemia, which is a very similar. But if this all this the liver function tests are normal, then you can suspect some sort of other find some sort of other disease, and the differential diagnosis of this jaundice. And if the other your blood test is normal, your bilirubin is normal, your liver function is normal, then you can think for the carotenemia. Then which is not a common disease, Addison's disease and anorexia nervosa, sometimes it may happen. If the increased total and indirect unconjugated bilirubin, what happens? So unconjugated hyperbilirubinemia. So unconjugated hyperbilirubinemia may be evaluate for hemolysis, peripheral blood smear, direct antibody test, which is Coombs test, and there is a one enzyme, glucose 6-phosphate dehydrogenase enzyme testing. These are important. If hemolysis present, treat underlying cause. If hemolysis not present, consider Gilbert syndrome or the Crigler-Najjar syndrome. Okay, because the Gilbert syndrome and the Crigler-Najjar syndrome is associated with unconjugated hyperbilirubinemia, where you are not finding any evidence of hemolysis, means your peripheral blood smear is normal, your direct antibody test is negative, and the glucose 6-phosphate dehydrogenase enzyme is negative, then you look for the cause, and the cause is one is Gilbert syndrome, another one is Crigler-Najjar syndrome. If you are finding the unconjugated hyperbilirubinemia, or you finding the indirect hyperbilirubinemia. The last one in next section, you see here the increased total means the total bilirubin is increased, or direct conjugated bilirubin level. So in total level or direct conjugated. So conjugated hyperbilirubinemia. Lab assay for the viral hepatitis like HBsAg, anti-HCV, and HAV, means hepatitis A. These are the three important tests. We routinely done this test, and the consider imaging. So the imaging is ultrasonography is a preferred first line test. So the viral hepatitis, if the viral markers are positive, then it is a viral hepatitis. If in ultrasound, if this gallstone is there, then you treat for the gallstone. If no diagnosis found, consider additional studies on the clinical suspicion, additional cancer testing, biliary imaging, and autoimmune antibody assay, where which you know that there is a one hepatitis, that is autoimmune hepatitis, and in that condition you should go for the autoimmune antibody assay. The same thing almost here. I would not repeat the same thing. Almost which I discussed earlier. I am skipping this slide. Here there is two gentlemen. You see that there is a Najjar and a Crigler. In 1953, Crigler and Najjar described a mysterious illness that caused jaundice and severe neurological damage. Here, the inherited disorder of bilirubin metabolism. This is another section of this my talk. Here, the one is Gilbert syndrome. The Gilbert syndrome is characterized by the unconjugated hyperbilirubinemia with the serum bilirubin less than 3 milligram per deciliter. This is very, very important. Associated with defect in bilirubin uptake. So, what is the defect in the Gilbert syndrome? There is a defect in the bilirubin uptake. The mutation in the promoter gene of UTGA1A, which is responsible for for this Gilbert syndrome. The fasting cause an increase in serum bilirubin. Why? Because the fatty acids compete for the conjugation with albumin to bound to albumin. So actually the bilirubin and fatty acid both compete to bind with albumin, and that's why fasting causes increase in serum bilirubin. Liver enzymes are within normal reference interval. In the Crigler-Najjar syndrome, the Crigler-Najjar syndrome are of two types. The Crigler-Najjar 1 and Crigler-Najjar 2. The Crigler-Najjar 1 is characterized by the complete deficiency of enzyme uridine glucuronyl transferase, leading to unconjugated hyperbilirubinemia. The serum bilirubin A level above 20 mg per deciliter. The unconjugated bilirubin above 20 mg per deciliter crosses the blood-brain barrier and causes kernicterus in neonates. Okay, so the kernicterus is what? Where there is a severe hyperbilirubinemia and the blood-brain barrier of neonates is not mature enough to stop entering this bilirubin in the blood-brain inside the blood-brain barrier, and that cause permanent damage of some brain cells, and that may lead to the mental retardation and certain other mental deformity. The diagnosis is based on the bilirubin greater than 20 mg per deciliter and normal liver enzymes. Then the Crigler-Najjar syndrome to the partial deficiency of enzyme uridine glucuronyl transferase, resulting in increase on conjugated bilirubin, which is in the range of 5 to 20 mg per deciliter. The unconjugated bilirubin is lowered upon administration of phenobarbital by inducing UGT enzyme. All liver enzymes parameters are normal and kernicterus is not found here. Why? Because kernicterus is possible when the serum bilirubin level is more than 20 mg per deciliter. Then it crosses to blood-brain barrier, and the blood-brain barrier of what? The immature blood-brain barrier. That is important. Then the Dubin-Johnson syndrome, the conjugated hyperbilirubinemia due to defective MRP2 protein, and this MRP2 protein is multi-drug resistant acting protein. Here this MRP2 protein is responsible for the secretion of bilirubin to bile caniculi, and here the liver enzymes are normal. What is the Rotor syndrome? They characterize the conjugated hyperbilirubinemia due to defective secretion of bilirubin to bile caniculi, but the etiology is not known. The normal liver enzymes here. So the summary: the bilirubin out by the hepatocytes involve 4 steps. The process involve molecules. Here you see the 4 steps are finely balanced. The first is the uptake, storage, conjugation and excretion. Uptake, there is no molecule still identified. Storage, GST is ligandine like molecules are responsible. In conjugation, the UGT1A1 is recognized as a molecule, and in excretion, MRP2 is recognized as the molecule. The 4 steps are finely balanced, therefore reduction at any step may cause hyperbilirubinemia. Enhancement of throughput require induction of multiple genes, probably coordinated by nuclear receptors such as the constitutive androstenedione receptor. So here I am concluding my this topic. Thank you for your attention.